Welcome back, friends. So welcome again in another video lecture from my YouTube channel. So before going to talk about today's lecture, I want to suggest you that if you do not know anything about what enzyme linked immunosorbent assay or ELISA is, then please go back to my YouTube channel and there will be a video mentioned the term named as enzyme linked immunosorbent assay explained within 10 minutes, and go watch it so you can learn the basics about ELISA and then come back to here. Otherwise you cannot correlate with ELISA and that can be a problem. Okay. And if ELISA, if you know so to well about enzyme linked immunosorbent assay, then let's get started with ELISA. Before going to talk about ELISA, I want to tell you that ELISA is simply a modification of standard modification, or you can say a one type of sandwich ELISA. Okay, in sandwich ELISA we sandwich the antigen between a captured antibody and the enzyme linked secondary antibody, and by adding substrate we will convert in presence of enzyme. By adding substrate we will convert those substrate into a colored product, and this colored products in colored intensity is measured with the help of spectrophotometer, by which we can conclude the concentration of antigen present. Okay, so that is the funda of sandwich ELISA. And in ELISA the only provide us whether antigen is present or not with the help of a small dot, or you can say blue spot. Okay, that is why in that experiment is named as ELISA. So I am not going to talk about in very details about ELISA, otherwise the video will be so long. I am not going to do that. I will mention you the steps that is necessary to perform a ELISA technique, and along with I will going to explain the necessary ingredients needed. I am not separately tell you about the ingredients during my explanation of the steps. I will talk about the details equipments that are needed. Okay, so that the video will be in brief and will be in a nutshell, right? So let's start it. So first of all, we will take a tube, or you can say a vial, where we will add 1 ml of 1x assay buffer and 50 microliter of serum sample, and we will mix it thoroughly. Now there is a question, that is, in kit they will provide you 10x assay buffer. Okay, so you need to convert it into 1x. Then how can you convert it? You will convert it in simply that way. You will divide it as 1 by 10 and then the amount in which you will sum up the total sample. Okay, so 1 by 10 into 1, so we have 0.1 ml. So 0.1 ml will be your assay buffer from 10x assay buffer, and you will add 0.9 ml distilled water with that. Okay, so if they are telling you that you have 10x assay buffer and we need 1 ml of 1x assay buffer, then you will simply add 0.1 ml assay buffer and 0.9 ml of sterile distilled water mixed them, and you will have your 1 ml of 1x assay buffer. Similarly, if they are telling you that you have 10x assay buffer and we need 10 ml of 1x assay buffer, then you will simply do this in such a way. You will divide 1 by 10 and you will multiply with 10, and you will have 1 ml. That means you have to add 1 ml assay buffer from that 10x assay buffer and add it with 9 ml distilled water, right? So I hope it is clear how to convert a 10x assay buffer into 1x assay buffer with the help of simple calculation. Okay, and this 1x assay buffer is mixed with 50 ml of the serum sample, and we will mix it thoroughly. Now come here. If you watch my previous video, then you know that during enzyme linked immunosorbent assay we will coat our antigen or antibody in a microtiter well. Okay, but in case of ELISA, we will coat or antibodies, we will coat, or our coated antibody or antigen will present ready-madely in a thing that is what we call ELISA strip. right? In ELISA strip there are three different areas. In one area it is our negative control zone, where it is completely filled with inert protein. Okay, there is no antigen or no antibody. In test sample, where we have to find out whether it is present or not, there will be immobilized antibody. Okay, and in positive control zone there is immobilized antibody along with antigen. So we have prepared three different areas in three different perspectives. One is for negative control, where there is no immune component. Another one is positive control, where there is immobilized antibody and antigen attached, and another one is test sample, where there is only immobilized antibody. So when we will add our serum here, then the immobilized antibody will bind with the antigen, and then when we add the secondary antibody conjugated with enzyme, we will have our spot, right? So one thing is clear: in positive control zone, no matter whether the result is positive or negative, we will always have the spot, right? Now let's move. So in ELISA strip, we will dip our ELISA strip in the tube where we have made 1 ml of 10x assay, 1 ml of 1x assay buffer and 50 ml of serum sample, and we will allow the reaction to occur for near about 20 minutes. Okay, that what exactly what I have written here. Insert ELISA strip and allow the reaction to occur for 20 minutes. After 20 minutes, we will wash the strip by simply dipping our ELISA strip in another fresh tube containing 1x assay buffer. Okay, that what I have written here, containing 1x assay buffer, which is of 1 ml. So what we have done, we have simply used the assay buffer for washing purpose, right? And we will dip this strip for one of the strip in 1 ml of 1x assay buffer for 5 minutes, and after each 5 minutes we will replace the buffer. Okay, in that way the washing per step will be done. Then after washing is completed, we will dip again dip the strips again in a vial or tube consisting of 1 ml assay buffer and 10 microliter of antibody horseradish peroxidase conjugate. I have told you about what is horseradish peroxidase in my previous video. If you have not watched this video, then go and watch. After this, we will allow the reaction to occur for near about 20 minutes. So what we have done, we have taken a tube or vial where we put 1 ml of 1x assay buffer and 50 microliter of what 50 microliter of serum samples, right? Then we will dip our ELISA strip there, and we will allow the reaction to occur for near about 20 minutes. After reaction, we will they kept our ELISA strap, and then we will dip it in a fresh tube containing 1 ml of 1x assay buffer for near about 5 minutes for washing purpose, and we will do it thrice, and after each 5 minutes we will replace it, okay, with a new buffer, and that is why the washing step is performed. After that, we will dip the strip again in a fresh tube consisting of 1 ml of 1x assay buffer along with 10 microliter of antibody HRP conjugate, that is our secondary antibody, and we will allow the reaction to occur for near about 20 minutes. Now we will wash the strip again by the same way which I have mentioned in our step number 3, right? That means we will we will take our ELISA strip from the tube, and then we will dip it in a fresh tube or vials consisting of 1 ml of 1x assay buffer for 5 minutes each, and after each 5 minutes we will replace the used buffer with new one. Okay, in that way we have performed the washing step once again, and then we will observe what is going on. So here I have mentioned if it is positive then what will going on, and if it is negative then what will going on. So remember, I have mentioned you that there is three zones. One is negative control, another one is test zone, another one is positive control. In negative control, I have told you that there is nothing, only there is inert protein. So if the serum sample contain antigen, it will not bind with this. As so, one thing is clear, no matter whether it is positive or negative result, we will not have any spot in both positive and negative. We will not have any spot in both the cases when we are considering negative control zone, and that is the reason why both in case of positive result we have nothing here, and we have nothing here, and in negative result we have nothing here. Okay. So, and in test zone we will have our immobilized antibody, and when we add the antigen simply bind with the FAB portion of the antibody, and then when we add the 10 microliter of antibody HRP conjugate, that is our secondary antibody, then when we add the substrate, here substrate is tetramethylbenzidine along with H2O2, where tetramethylbenzidine is the chromogen and H2O2 is substrate. And how this reaction is going on, I am going to talk about a little bit after. You don't need to bother about that. Then after adding the substrate will convert this into a colored product. Okay, and this tetramethyl benzidine oxide will produce blue spot. And in positive control zone, as I have previously told you, no matter whether the positive or negative result is, we will always have blue spot in case of positive control zone, as it has already its antigen. Okay, so secondary antibody will bind with it and provide the blue spot. Okay, so I think that you have figured out what will be the spots will be. Okay, so in case of negative control zone we have nothing, in test control zone we have blue spot in case of positive result, and in case of positive control zone, no matter whether the result is positive and negative, we will always have our spot, right? So in case of positive result, we have two spot, one is in case of test zone, another in case of positive results, positive control zone. And in case of negative control zone, we have no spot. In case of, let me zoom it, now that will be the, in case of negative control zone, we have no spot in our test zone, and negative control zone will not have any spot in both the cases, and we will have positive control zone in, will have the spot in case of positive control zone. Okay, so that is the difference between positive and negative result. Positive result will give us two spot, whereas negative result will only give us one spot. Now let's move to the how the reaction is going on. So when you add the second substrate, here substrate is H2O2. H2O2 react with horse-radish peroxidase, and horse-radish peroxidase release its oxygen from H2O2 and convert it into water, and this oxygen will oxidize tetramethyl benzidine into tetramethyl benzidine oxide, and it will give the blue spot, and that is the idea. So I hope it is clear, and if you find it helpful, then like the video and share with your friends and subscribe my channel. Thank you.